That's the basics of replication. So now we're going to get into more of the details of the specific proteins that are involved and the steps that are involved in replicating the DNA. So it doesn't just automatically happen, right? There's always proteins that are going to have to facilitate the process. And it's a pretty complicated process. So on this one, what I'd like you to do is I've got for all of these, if you happen to have the handout, these are already listed on the handout, but try to cover up that handout and then see if you can kind of reason out which ones are going to go with which. And then if you feel confident about that, then you can go ahead and start figuring out which numbers in the image are associated with the particular proteins. And then if you want to check first, so what I will do is I'll give you time to pause, and then I'll come back and I'll make sure you have these definitions in the right spot, and then I'll give you another pause once you have the definitions checked to then go and see if you can label the structures. So first pause and see if you can move these around and see if you get them in for the right function for each particular component. Okay, and so to start, so the RNA primer is the thing that is going to provide the beginning nucleotides. So the RNA primer is just a few RNA nucleotides that primes the DNA replication. And I don't know why it can't be a DNA primer, but it has to be an RNA primer. Okay, thanks, Dylan. Okay, can you give me a little bit? Okay, thanks. My 4-year-old is just telling me that lunch is ready. Okay, so the RNA primer, it is, like I said, I don't know why it can't be a DNA primer, but it's got to be an RNA primer to get it started. And it might just be because we don't have a DNA primase. It might just be, that's my guess, is because the thing that synthesizes the primer is an RNA primase. Anyway, so the RNA primer is going to begin the replication by providing the nucleotide, the beginning nucleotides. The replication fork is just the spot where the DNA strand is separating. And then the primase is the thing that synthesizes the DNA primer. And then DNA, so once it is separated, once the DNA is separated, you have this short piece for a little while that is single-stranded. And single-stranded nucleotides or single-stranded nucleic acids are unstable. So there are proteins that can come in or enzymes that can break the single-stranded DNA. But if you have single-stranded binding proteins on there, they are going to stabilize the parental strand while it is in that single-stranded state. The helicase, if you think about with the enzymes, right, the primase makes the primer. The helicase is breaking down the helix. So that is what unwinds and separates the parental DNA. Topoisomerase. Now this one's a difficult one, but if you remember isomer, right, you're changing the structure a little bit. So that topoisomerase comes in when the DNA is double-stranded, because if you think about a helix and it's twisted, right, if you come in and you take it and you grab it from the top and you start unwinding it, then there's going to be stress on the bottom, right, and it's going to break and it's going to become a tangle. And so what the topoisomerase does is it comes in and it cuts the double-stranded and then twists it to relieve the strain. And that prevents the strands from just breaking. And then the DNA polymerase 3, it is not shown, but that is the enzyme that's going to come in and add new DNA nucleotides. And so when you have the definitions, it might be easier to come through here and then label the components. So if you haven't done that already, pause and see if you can get the right components with the right numbers. So the RNA primer, right? This is the thing that is going to get it started. And if you haven't already, I want to orient you. If you could, in this chain here, just circle one nucleotide, and then I'll do it as well. But one nucleotide, if you were to circle one nucleotide here or on your worksheet, what would you circle? Just think about it for a second while I get my pen going here. So the way this is represented, each one of these little teeth right here is a nucleotide. So this is a nucleotide, excuse me. Right, so each one of these is a nucleotide. And between, just as a quick review, right, what bond is between the nucleotides within one strand? Right, a phosphodiester bond. And then what kind of bond is connecting the nucleotides between two strands? Those are hydrogen bonds. Good. And so once we have started separating it, right, the thing that is going to get things started is going to be that RNA primer. And so in this one, this RNA primer only has two nucleotides attached, right? But now it's hydrogen bonding to the parental strand, and then now we can start adding the nucleotides here. So that RNA primer is number 4. And I'm going to stick in this region, so looking at this number 3, right, this number 3 pink Pepto-Bismol thing here, is the thing that is making this primer. And the thing that makes that primer is the primase. So this number 3 is primase. And then right along here, where the DNA is separating, right, that is the replication fork. So number 6 is this place, and that is the replication fork. And then after it has, well, here, let's look at this enzyme real quick. So this enzyme here, it's like the little zipper pull, right? So it is, you're pulling your zipper, that is unwinding the, disconnecting the two strands of DNA, right? So that is unzipping, so breaking the hydrogen bonds between the two strands of DNA. That is the helicase. And then, once that helicase comes through, and now you have this little section that is single-stranded before you start getting the new nucleotides added. So it's actually not very much of the strands that are single-stranded for very long, but you need to stabilize those. So these little tic-tacs here are the single-stranded binding proteins that are going to stabilize the single-stranded DNA. And then this chunky protein that is functioning ahead of the break, or ahead of the helicase, is breaking and then twisting the DNA to relieve the strain. So this is that topoisomerase. And then just think about if we were to draw polymerase 3 here, that's the thing that is going to add new nucleotides. It's going to add it always at the 3' end. And so that DNA polymerase 3 is going to be located right here at the 3' end of the RNA strand. And so if we go back and label our 5' and 3', really you can only label it of this little strand here. right? The 5' is here, and where the arrow is the 3', so that's where the new ones are going to be added.